Because otherwise it creates, if you just do streaming on YouTube, it just creates a new like YouTube live event.
And that's why it wanted me to connect to a YouTube account.
Yeah.
Okay. All right.
Okay. In general, for future reference, I've preset the meetings to have the right YouTube credentials. So,
so that's all I have to do.
Yeah.
Perfect. All right, I'm going to close these rooms and bring them back.
Yeah, because you have to base. It's actually annoying because you have to set them all ahead of time, because otherwise they default to the last one that you set.
Yeah.
Per meeting. So for like all recurring meetings, I'll try to link to the same thing.
Okay. And then do you set it on speaker view? So that's the only thing that's okay.
And if you want, you can pan or spotlight a video, and it'll be sort of by default the one that's shown.
Let's see. So, oh, everyone's back now. It's hard being on speaker view. I had no idea that there were even other people around.
Yeah, that's why I have like 4 screens going at once.
Yeah. So go ahead, Abby. Everyone's back.
Everyone's back. Do you guys have any questions before I start going through this final slideshow? All right, yeah, again, just you can stop me as we're going along. So, okay, so we wanted to look to see, or you guys already identified mutations, most of you, in that kind of were the cause of these wrinkly phenotypes in the mutants. And what you should have found is either 1 or maybe 2 mutations that occurred in one of these 4 genes. So either WISP-A, WISP-F, YFIR, or MOR-A. And some clones were double mutants, so they actually either had a mutation in 2 different genes or had 2 mutations in the same gene. So how are these important? So all the mutations we saw come from 3 pathways that we know are important in increasing a secondary signaling molecule called Cyclic-di-GMP. So this is just a really broad overview of looking at the pathways, but what you should take away from this just is that they're important in increasing Cyclic-di-GMP. So why is Cyclic-di-GMP important? Because it governs this decision in bacteria of whether they should stick to a surface, produce biofilm, or swim away, going through dispersal to kind of start a new bacterial population. So if cyclic di-GMP levels are high, that's going to be associated with high biofilm production and low motility. And if they're low, that's associated with bacterial dispersal. So cyclic di-GMP is, its production is catalyzed by diguanylate cyclases, and it's then degraded by phosphodiesterases. And why I'm pointing this out is because within these pathways, all of them contain a diguanylate cyclase. So the WISP or Wrinkly spreader pathway is essentially an elaborate antenna system that senses cell contact and then transduces a signal that will activate a DGC WISP-R, and then that's going to increase production of cyclic di-GMP, and then which then increases production of biofilm polymers. So this is just looking kind of at the specific mutations that we saw. So we saw mutations in either WISP-A or WISP-F. And these are important because WISP-A is methylated after sensing the signal. And this is what starts this signaling cascade that results in the activation of the WISP-R DGC. WISP-A methylation is going to cause it to be more receptive to the input signal. And then this whole process is negatively regulated by WISP-F because it removes that methyl group from WISP-A. So the mutations that we saw, how they're affecting the typical regulation of this pathway, is that we saw either mutations in WISP-A or WISP-F. And what we think that they're doing is causing WISP-A to be essentially hypermethylated. So it's continuously on and then continuously activating WISP-R, increasing cyclic di-GMP production and increasing biofilm formation. Do you guys have any questions on that? Okay, so the next pathway we'll look at is this YFI-BNR. So this is also, sometimes they have different names, known as the alternative Wrinkly spreader pathway. So in this case, YFI-N is the diguanylate cyclase that's going to activate increased cyclic di-GMP production and increased biofilm formation. And it's negatively regulated by attachment of YFIR. So YFIB, in response to membrane stress, will actually sequester YFIR. And then that allows for this increased cyclic di-GMP and increased biofilm production. So where we saw mutations were in YFIR. And we're thinking that that's interfering with its ability to negatively regulate YFIN. So then again, YFIN's more active, increased cyclic di-GMP and increased biofilm production. And this third pathway is the MORA motility regulator, so also known as Mike's Wrinkly spreader. So this is just a single protein, and it contains an active DGC and PDE, a like degradation domain. So it's able to both produce and degrade cyclic di-GMP. And what we think is that these mutations that we identify in this gene are resulting in increasing its DGC function. So then again, increasing cyclic di-GMP production and increasing biofilm formation. Does anyone have questions before we just talk a little bit about relevance? So, so how are these studies? So I'm just going to talk about one way that they can be really directly relevant to looking at Pseudomonas aeruginosa. So Pseudomonas aeruginosa is a major opportunistic pathogen. So it infects people with cystic fibrosis, people on ventilators and burn victims, many other people that are sick. And the WISP pathway is nearly identical in Pseudomonas aeruginosa and the bacteria used in the classroom experiment, Pseudomonas fluorescens. And we find that Wrinkly mutants often evolve during infections from Pseudomonas aeruginosa. So here's just one example that came partially from our lab, where they established, in this case, a burn wound on a pig. and then inoculated that burn wound with a mixture of different Pseudomonas aeruginosa strains and found high biofilm forming wrinkly mutants pretty quickly into the experiment, and they persisted throughout, so for almost a month. So these wrinkly mutants had elevated Cyclic-di-GMP, and that caused this high biofilm phenotype. And then performing whole genome sequencing, just like you guys did, in this case on Pseudomonas aeruginosa, showed that the mutations that cause the high biofilm phenotype were exclusively found in WISP-A and WISP-F. And that's kind of the whole from beginning to end. So from experiment to genome sequencing, to just thinking about why those are important in the bacterial cells, and then why understanding these kind of studies might be important for medical applications. So I'm just gonna stop sharing, then see, what questions you guys have after going through the whole experiment, or anything you'd want to talk about the last couple minutes of our week.
I clicked on the survey, and for the questions that ask for a written response, when I click on them, it doesn't let me type. Am I doing something wrong?
This was my first time setting up the survey, so probably it was me. And I don't know if you can help with this, David.
I will have to look at it to see. Let me try to look at it very quickly to see if it. Let me see. Why don't I try to take it and see if the same thing happens to me? Abby, do you want to tell them, again, to be honest in the survey and what you're going to use it for, and why they should help you by completing this?
Yes, please complete the survey. Be as honest. Yeah, be completely honest. So the hope is, you guys, is the so I'd like to extend again. I know I talked about this in a smaller group, but I really appreciate you guys going through this module with us, because it's the first time we're really testing it with students. And I think that this has been a really great way to see, what works better or worse. But I'd love to get your exact opinions to kind of, what did you like? What did you not like? How can we improve to help roll this out in classrooms, especially because I feel like, at least for the next year, students aren't going to have the opportunity to do hands-on in-class experiments. And so hopefully this is a way for them to get some exposure to that without that classroom experience. So that's a goal of ours. And this, I think your feedback will be really useful in refining the curriculum to hopefully make it more fun and that more engaging, so that people want to do it and feel like they got something interesting out of it in the end.
Okay, so there was a problem, Abby. Whenever you set up the open-ended questions, you did it as basically a graphic rather than a text entry. So everyone, I hope you haven't already done the survey. I'm going to send another link. Oh, actually, it's still the same link. So maybe it will work now if you refresh. We'll see if it's saved. Yeah, it should work now. But just take, 5 minutes now, 5 or 10 minutes now to do this. Or if you have other comments, feel free to tell Abby. I'm sure she'd love to hear it.